get too many copies. So how is that precisely managed is the fifth one that we'll talk about. The final one is accuracy. And accuracy, as you can imagine, is crucial to ensure that we don't accumulate mutations every time we make a copy. So this is the problem of how you go from AGTC to only AGTC. So have these as sort of anchor points that we'll come back to throughout the lecture to make sure that you've discussed each of these aspects, and at the end of this lecture you'll be able to understand how each of these steps are achieved, and at least in principle and in outline. Throughout the lecture I'll tell you at what level of detail you should remember for the exam, and I'll also tell you at what level of detail you need to have an intuitive understanding for what's happening. Not just that, oh, there's some molecule that's here, but what does that molecule do? Okay, so there are 6 problems that we'll discuss. And at the end, you can imagine accuracy is a problem not only when you copy, but also when you just have a cell with DNA in it. If the DNA can be mutated, if the DNA is not accurately copied, can you repair it? Okay? And repair mechanisms are absolutely fundamental for us surviving in the environment. Every time you have the sun's UV radiation or anything that hits you, you can have mutations that arise, which then have to be repaired. And this is so fundamental that earlier this month, I think, or last month, it was announced that the Nobel Prize for Chemistry was given to DNA repair. Okay, so the later third of the lecture that we'll talk about was this year's Nobel Prize. I think the lecture hasn't yet come out, but the news is out that this is the problem for which the Nobel Prize was awarded in chemistry. Okay, so why all these problems of polarity? An important aspect that you have to remember is the way the cell solves is by using the same machine that always makes DNA replicates in the 5' to 3' direction, but it does so slightly differently on both strands. The direction is still the same, okay, and we'll figure out how that coordinated replication happens. As you might imagine, prokaryotes and eukaryotes have slightly different problems. In prokaryotes, there is no nucleus. In eukaryotes, there is a nucleus. Not only that, prokaryotic DNA is circular. Eukaryotic DNA are long chromosomes. Okay? So because it's linear, there are some further challenges that eukaryotes have to deal with that prokaryotes don't have. Another reason for challenges in the eukaryotes is there are histones. The DNA is wrapped around histones, so every time you replicate DNA, you're making twice as much DNA. You now need to have twice as much histones, and you have to make sure that those are replicated. So we'll talk about those challenges. And then we'll talk about DNA repair in the context of this amazing accuracy with which replication routinely proceeds. 1 in 10 to the 9, okay, which is an amazing accuracy. And we'll also talk about how mutations get fixed. So this is sort of the overview. As we go through, I'll take you through each of those steps. So first off, how many of you have heard of semi-conservative DNA replication? Okay, some effort throughout. So that's the mechanism by which DNA replicates. And it turns out that the experiment that discovered this, does anyone know the name of the experiment? Ever heard of it? Messelson? Did I hear that? Messelson-Stahl experiment. Right. So Messelson and Stahl were 2 guys who figured out how DNA replicates, and that the replication mechanism is semi-conservative, meaning if you started with DNA that is yellow here, then the new copy is made by keeping 1/2 the same and having the second one new. Okay? This experiment is so famous and so good that if you Google the most beautiful experiment in biology, this is what will come up. So I'll let you do that at your leisure and figure out what that is. Messelson is still around. In fact, he was 1 floor above 5 years ago when I was a postdoc at Harvard. So he's still there doing active research. So all of this has been figured out fairly recently. If you start with one copy and you keep replicating, this immediately raises problems and solutions that you can imagine. First one is if you have a damaged DNA by accident, then because you're replicating, you'll keep getting many more copies of that mutation. So you need to fix pretty quickly, right? So if you say, let's just go a few divisions and then fix the mutation, that's not going to work, because the process of replication will preserve whatever change happened. The second problem is that the copying errors that you make as you replicate also will be preserved if you don't fix it before the next copy. Okay? So there are many reasons why the repair has to be tightly coupled to when you replicate, and it has to happen immediately before any one replication event happens. So we'll talk about why those things occur. All right, so the fundamentals, I can't stress enough. The replication always happens in the 5' to 3' direction. Let's give you a refresher on how DNA looks. The 5' refers to the phosphate, okay, that's on the sugar. And the 3' refers to the hydroxyl that's on the sugar. You can see that replication is going in this direction by the addition of a new base in the TP form, okay, CCP, which is then getting added to the hydroxyl. So the direction is 5' end to the 3' end. Okay. Throughout in all my slides, I'll tell you the important things that you need to remember will also be written in the slide. If it's not written on the slide and I'm saying something and you want me to slow down, please tell me to slow down and I'll be happy to slow down and discuss more in detail. Okay, so the important thing to remember here is replication happens in the 5' to 3' direction. New nucleotides are added at the 3' end, chain growth is in 5'. So another thing that's schematized here in pink is that there must be a piece of DNA already there, or RNA already there for you to replicate. So that's called a primer, and a primer is required. And as you can see, it's because of this direction of one strand going 5' to 3' top to bottom, and the other going 5' to 3' bottom to top, that you get the anti-parallel helix structure of DNA itself. Okay? All right, so the replication machinery that the key protein that does this is called the DNA polymerase. We won't go into crystal structures or anything. The crystal structure exists. If any of you are into playing with RASMOL or any of the molecular dynamic software, you're welcome to do that. But for this course, all you need to know is the structure of the protein looks a bit like a hand. And so it's referred to as such in your textbook as well. There are the fingers domain, the palm domain, and the thumb domain. Okay? So you can think of it as DNA is just going through that outstretched hand. And as it's going through, a new nucleotide comes in as a triphosphate. Its position leads to a hydrolysis, which then leads to the incorporation of the new nucleotide, and then keeps going, the chain keeps going forward. Okay? So all you need to know is that there are fingers domain, palm domain, and the thumb domain, and it's literally because the whole protein looks a bit like an outstretched hand. And the 5' to 3' polymerization itself only has an error rate of 1 in 10 to the 5. Now you remember that the accuracy that I mentioned earlier, that was 1 in 10 to the 9. Okay? That's the rate at which it makes mistakes. Only 1 in 10 to the 9, which is not accounted by just the polymerase. Just the polymerase only gives you 1 in 10 to the 5. We'll talk towards the end of the lecture about how the 1 in 10 to the 9 is achieved. Okay? The second problem, going back to this polarity that comes up, is if you have two strands, one that's going left to right, 5 to 3, and the other that's going left to right, 3 to 5, and yet replication must happen always 5' to 3', it must happen differently in the two strands. Okay? And it's being done by the same enzyme as we saw before. And that's shown here. We call that the leading strand versus the lagging strand. How many of you have already heard of leading strand and lagging strand? Okay, quite a lot. Great. So the level at which we are going to talk about in this lecture is the level at which you need to know for the course. I'll dive right in to the critical aspects. You already know then that there are two different kinds of replication mechanisms that must happen. One of them, the polymerase just needs to start at the 5' and keep going. So that's 5' to 3' continuous. That's the leading strand. The lagging strand cannot be replicated in that way because the 5' end where it can start is still base paired and occupied. So the way that's achieved is by making small fragments of primers called Okazaki fragments after small fragments of DNA from RNA primers. It's those DNA fragments that are called Okazaki fragments after the scientists who discovered it. And as you can see, this gives a solution for that first problem, the first problem of strand polarity. Okay? Because you synthesize one way on one strand and another way on the other strand. So for that synthesis on the other strand, and in fact in both strands, you need a primer, and that primer is given by a protein called DNA primase. And as you might imagine, this protein doesn't itself require something else, because if it required something else, nothing will get started. So this protein, all it needs is a single strand of DNA. It will recruit RNA primers that will go in the 5' to 3' direction, giving you a hydroxyl. So it's a primase, ACE for any kind of enzyme, prime for the primer. It just adds a short RNA primer. It's only about 10 nucleotides long. Okay? Now once that's added, you only have to remember what sorts of processes need to happen and read the whole series of steps at your leisure. The bottom line for what needs to be achieved here is you've added an RNA primer, now you make DNA until you bump into the one that's ahead. Okay? And then some enzyme comes in, chews off the RNA. We'll talk about what that is briefly. It's RNase H, which stands for hybrid of RNA to DNA. And then you simply extend further with DNA polymerase and stitch it up with ligase. Okay? So you can think of this nick simply being sealed by ligase. So what I told you is again written in the slide as such. Each Okazaki fragment catches up to the one that's ahead. RNase H removes the RNA primer, and then the nick between the two strands gets sealed by a DNA ligase. So if I were to ask you how does lagging strand get synthesized, these are the things that you might want to remember. Now, the ligation reaction itself, we can think about it in greater detail because it shows up again and again in many different contexts. And every time a DNA ligase comes in and does this ligation reaction, the mechanism is exactly the same. So we have this nick that's left behind. There is, again, a 5' phosphate and a 3' hydroxyl. ATP is used in this reaction. The first step is to make this ATP intermediate. This is a high-energy phosphate. ATP is getting used. And after that, the hydroxyl just seals the gap by a nucleophilic attack, leaving AP out. Okay? And then you have the fully ligated DNA. So as I said, it proceeds through the covalent linkage of an AMP. That's why you get this APP intermediate. And then you have hydroxyl attack and release of AMP. All right. So that brings us to problem number 2, which is how do you unzip the DNA? It's a very long, highly base paired molecule, so you can imagine you need a lot of energy to do it. And you use ATP to achieve this. The enzyme complex that does this is called a helicase. It'll become clear why it's called a helicase. It literally moves around an axis like a helix. And here's what it can do. If you took this enzyme complex, put it in vitro in a test tube, gave it DNA that looked like that, a long piece with another piece that is base paired, then gave it ATP, it can propel through, much like a propeller, unzipping the DNA as it goes. Okay? So it is uses ATP, remember that, has to use ATP. And it's typically made of 6 subunits, which we'll discuss in the next slide. These are those 6 subunits. As I said, the movement occurs through the hydrolysis of ATP. So you can imagine if it is like a rotary engine that's moving forward, and it has 6 different units. For the units to spin, first of all, not all of them have to be the same. You have to get energy from doing something in the engine, right? So an easy way to think about what's going on is to look at these 6 subunits, 1, 2, 3, 4, 5, 6, and look across from each subunit. You have pairs of subunits that have ATP, and the next pair has ADP and PI, and the next pair is NP. So what's happening is it binds ATP, a pair of subunits bind ATP, hydrolyze, and that hydrolysis shifts them over to the next position, and then release of ADP and PI shifts them over to the empty position. So it keeps spinning in the plane of the screen. And that's how you have unzipping reaction happening. Now, if you unzip a lot of DNA, then you have a lot of single-stranded DNA that's generated. So if you say this did the unzipping, you have a lot of single-stranded DNA that's generated. And when you have single-stranded DNA, it can itself anneal anneal with itself to form short base paired structures. And when the polymerase comes along to then replicate, that could be a hindrance. So there is a solution for that as well in biology, and which are these single-stranded DNA binding proteins. They bind without having any sequence specificity. So it doesn't matter what the sequence of the DNA is. They literally hold on to the phosphate backbone along the way. They don't care what the bases are. So that literally straightens out the DNA so that the advancing polymerase can march through. Okay? So we've talked about polarity, we've talked about how unzipping happens, and the problem, a sub-problem of unzipping, where you need to make sure single-stranded regions remain single-stranded. Okay? All right. So the third problem then is processivity, which I said is if the polymerase gets onto one end, either leading strand, lagging strand, wherever you want to think of, the 5' end, where it's beginning to make in the 5' to 3' direction, how do we make sure that it doesn't fall off? Okay? Processivity refers to the ability of an enzyme to keep doing that for a long distance. The ability of DNA polymerase to keep doing that for a long distance. And that is solved by using something called a clamp. Okay? And what the clamp is, let's say, imagine this has moved forward for a while, and here's where the polymerase is. There is a circular protein complex, a ring-shaped protein complex that's behind, with the DNA in between. Okay? So it's as though the polymerase can't go back and can't fall off. So it's held like that and moved forward. And how is this clamp put on there? That's what's described here in this slide. What's shown here is how a sliding clamp is loaded. Okay? Sliding clamp is this ring-shaped thing, as I showed. As you might imagine, loading this onto DNA would require lots of conformational changes. You have to open the ring, put the DNA through, and close. And that's achieved by a clamp loader, which binds ATP, opens the ring, DNA goes through, ATP is hydrolyzed, and now the clamp is loaded. And now that the clamp is loaded, the clamp loader can fall off, okay? And can go back and cause another round of this to happen. Okay, so ATP is used to ensure that processivity is maintained, and clearly conformational changes are required for that. The clamp goes around and then makes sure that the polymerase stays on. Okay, and here's a picture of that. The two subunits are in two colors, and white and light green is the DNA going. So the things that we've talked about so far are only 3 of the problems, and yet they together form this big mess. Okay? So So if you haven't seen this before, this looks very complicated, which is why I'll put the video up again on the course website for you to play through and think about it. Here's the summarized version. Everything's happening simultaneously. You have a leading strand and you have a lagging strand here. Okay? That's the first thing, orient yourself. The red is the newly synthesized DNA. Don't look at all these machines just yet. The blue lines that are dotted, that's the RNA primer that's priming the Okazaki fragment to be made. Okay? So you can tell this is the lagging strand, that's the leading strand, new DNA is being made here. And the one with the arrow, that's the DNA polymerase. So you can see there are two different DNA polymerases that are being used, one for the leading strand and the other for the lagging strand at the same time. But they're all together in a complex because the helicase is necessary to go through and unzip. Okay, before that, neither strand can be replicated. So this yellow helicase is going through and unzipping and spooling off things on either side. In blue here is the DNA primase, which keeps putting down that RNA primer. And the clamp loader is constantly putting the sliding clamp on to make sure that things stay. Okay? The polymerase doesn't fall off. So you'll see this whole thing animated in the video, and I encourage you to go take a look at it. You'll get a better understanding. The one that I played at the beginning, you'll get a better understanding of what's going on. So here's an EM picture of the entire process. It literally looks like the schematic that you saw in the previous slide. So any questions so far regarding replication up to this point, the problems that we've discussed so far? Any questions? Okay, so if you're clear, we'll keep going. The next problem that we'll talk about is this problem of coiling up DNA. There are lots of cliched explanations for it. It's literally any ball of yarn that you can think of. If you straighten one part, then the part that's behind will get coiled up some more. Old telephone cables used to be used as an analogy for this. So here's the problem. You're trying to make more and more DNA on this side, but the base paired region, then it's getting really tangled up, so you need to quickly move it around. Okay? So you quickly need to unwind that. So that untangling, how is that achieved? That's what we'll talk about next. Here's a second illustration of that. You have the helicase that's just zipping through. There's coiling that's happening in this direction so that you get something like this, a coiled mess, that then needs to be uncoiled. And that's achieved by proteins called topoisomerases. How many of you have heard of this protein? Okay, half the class or something like that. So topo, you can think of that as shape, morphology, something like that. Topoisomerase. Isomer is just changing one thing, one form to another, but equivalent. So it's just something that changes the topology or how things are put together without really changing chemically what's in there. Okay, so topoisomerases relieve this torsional stress. And there are two kinds of topoisomerases, and both of them are used to do really similar things. We'll talk about both those mechanisms. The first one is a topoisomerase that unentangles by a mechanism called the nick and swivel mechanism. And the way to remember this is literally what those two words say. It makes a cut on one strand and then it moves it about. So if you can imagine these as two adjacent regions on DNA, the nick and swivel would be you open this and then you move it this way. Okay? You just spin around this axis. So that relieves the stress and it does not use ATP. So I'll show you how that's happening here at a closer view. These proteins have a tyrosine at their catalytic residue. The hydroxyl attacks the phosphate backbone. It gets covalently attached. So the topoisomerase enzyme itself is attached to one end. So in this hand diagram that I was showing you, it gets attached over there. and then swivels across the other backbone. Remember, the carbon-carbon backbone bonds can all swivel, single bonds